monitor calembula and a number of other things. I'll refer to that. And then as an add-on, we did some bee work that was fascinating to address the question of what do flowering cover crops do to pollination. Sorry, that is quite small. We had on the top, left to right, treatment one was a less diverse, almost pure grass cover crop in the work row. The second one, we added some medics. Third one, we added subterranean clover along with medics. And then the fourth one was our diverse cover crop that had buckwheat, which we didn't actually end up planting. That shouldn't happen, getting ahead of itself. Mustard, phacelia, and triticale was planted in all of the treatments at 20 kilos. Then under the trees, treatment one was basically compost and mulch. 2 was compost and wood chips. 3 was a green mulch with compost, and 4 was diverse. So it had subterranean clover, medics, mustard, etc. in it. If anybody wants that recipe, you're welcome to it. The parameters that we measured, and part of this trial was what actually and how do you measure. So I'll go through them very briefly. Cover crop performance in terms of how they did in those environments, plus a bunch of small-scale trials. Biomass production, how much did we actually produce per square meter. Soil analyses, it was a Haney test, plus others via NemLab originally to Ward Labs in the States that were absolutely immense. Nematode biodiversity via NemLab. Bacterial fungal biodiversity through Sporotech on campus. Plant biodiversity, what actually happened during those 4 years. There was a succession of weeds that came through and things did well and other things went away. Litter breakdown. Calembula biodiversity. Calembula are very primitive insects that are involved in litter breakdown amongst others, and there's quite a diversity of them. And then bees and cover crops were measured. The soil quality parameters, it's just this vast volume of stuff that's very difficult to present. But I think the treatments, I think the student statement here is very relevant. Treatments had high values for soil quality parameters, with the exception of NH4, and soil food web structure indices at the end of the trial than they did at the beginning. That sums it up. Fixed carbon in soils didn't increase as much as I would have liked. Stability did, and so did diversity in some cases. The calembula, very difficult to count. You have to be taxonomically skilled. Are there measures? Yes, there are. I think we can refine that, but we counted everything. Yes, some were correlated with litter breakdown and were reasonable indicators. I think as a group we can refine that. The last of the results, it was really interesting. Basically, we mowed out the flowers in our control blocks before blossom, and then counted bees amongst others on blossom. So it was blossom visitations. The take-home, the blocks with flowers on their cover crops had more flower visitation by bees than the mowed rows. So that dispels that myth, and it was replicated on the left in 2020, and then on the right, 2021, and the bar graphs on the right-hand side indicate flower visitation. So we had a substantial increase, a significant increase in blossom visitations. That was significant. Just to give you an idea of the calembula, there they are. They're really small. They're quite difficult to collect. You've got to put them in a funnel or whatever. The litter breakdown trials were run in parallel with those. Litter breakdown was fascinating as well. We used leaves out of the trees, so we weren't using another medium. I'd just like to thank Hortgrow and ZZ2. It really worked really well. I learnt an immense amount, and we had a lot of, it was very rewarding, and that's just a pretty picture in terms of what carried on, what initiated on that farm. That was full green cover crop, not in the trial. That was in second, third year. They sort of looked at what we were doing and then modified, and that worked really well. Thank you.